So as I said, my name's Steve Coleman. I'm from VT-iSIM. So we developed the iSIM system, and I led the R&D team on the initial development of the iSIM and then the future developments that have taken place. So what does the iSIM do in terms of application? You know, we don't design instruments to image beads or to image lines or separations. We design instruments to image biology. So it offers high temporal resolution, so imaging at 200 frames a second. You know, personally, I don't get caught up in specsmanship over, you know, what can we achieve on a bead, or I look at what can we achieve in real case scenarios, and you know what's been published. So here we're doing 200 frames a second at 1K by 1K, high spatial resolution, so imaging at around 100 nanometers in XY and around 270 nanometers in Z, and live cell imaging, so very long time-lapse experiments. Example, there's a 15-hour time-lapse experiment. We have some groups who would do several days without photo bleaching or photo damage to the cells. We also don't suffer the traditional limits of imaging depth with traditional SIM. The name is something I'll come to in a little while. And it operates as a simple camera-based imaging system. So you set your laser power, you set your camera exposure, and you just shoot. So what's the journey of iSIM and kind of my journey through the technology as well? Really, I think iSIM comes under the wider umbrella of image scanning microscopy. And you know, I only have 10 minutes, so I can't go into too much depth, but the basic principle of image scanning microscopy is that, you know, if we take a regular confocal laser scanning microscope and we detect onto an array detector rather than onto a PMT, for example, then we can take each pixel of the array detector or each cell of the array detector as being a very small pinhole, a micro pinhole, which allows us to image it at a higher resolution. But if we have multiple of these small pinholes, these micro pinholes, then we can detect all of the emission and we can reassign the image information to where it originated. And this allows us to maintain very high signal to noise and also allows us to achieve high resolution. And I could go back to Shepard and published on this on the 80s, but I think Muller and Enderlein was the first one that I came across. Harry sent me the paper back in 2011, I think, and we started on this journey. Obviously, probably the most well-known implementation is the Zeiss AeriScan system, which uses their linear spectral detector, which has 32 channels on it. Each is a micro pinhole, effectively, and then they can reassign image information to where it originated. There's the group in Genoa who've taken it further with the SPAD array detector and some really beautiful work there. But I would say that all of these techniques are kind of digital implementations of image scanning microscopy. You grab an image, you have your micro pinholes, and then you process it by reassigning the image information to where it originated. And this is really where I started. So, you know, Harry sent me the Muller and Enderlein paper. At the time, we developed a 2D array scanning confocal, which was similar to spinning disk, but rather than using a rotating disk to scan multiple points, we used the Galvo to scan multiple points. And then we looked at maybe we can use this Muller and Enderlein technique. It doesn't matter, in the top left, and we can multiplex it. So we can just do it with multiple points, grab an image at each scan position, reassign the image information, and try and parallelize that process. And this worked well, and also Harry and Andy also did an implementation using DMDs, which also worked well. But it's still limited in terms of the speed of acquisition, in terms of the photo bleaching that it caused. And really, that's kind of what led to a eureka moment in Harry's lab where they looked at this and thought, well, hang on, we don't actually need to reassign this image information through digital means. We can do it by analog means. So if we take the picture on the left there, so we have an emission point spread function projected onto an array detector, a camera. We can take each pixel of the camera as being a micro pinhole to allow us to have our higher resolution. And yes, we could take an image there, then reassign the image information to where it originated, or we could shrink the image of that point spread function onto the array detector, and we actually reassign that image information in real time. So now we don't need to do any post-processing. We can parallelize this process so we can go extremely fast, and we can also have all the other advantages of multi-point confocal scanning by having very low photo bleaching. So this is the 2D array scanner. So if we take out the emission microlens array, this was actually a 2D array scanning confocal device that we developed where you just illuminate a microlens array to create a 2D array of points. You scan them onto a sample plane. You descan the emission, come back through the pinhole so that you have a confocal image and rescan onto the camera. And the nice thing about this implementation was that everything from here on the emission path all the way back to the backside of the Galvo is effectively stationary. You're just relaying a multitude of points which you then scan onto the camera. Now, the big advantage of this in terms of implementing iSIM, Instant SIM, was that what we could do is actually we could place this reassignment optic, this emission microlens array, and isolate it into the emission path where we're just relaying a series of images. We're not doing any scanning. And then all of a sudden we have this very simple implementation where we scan our diffraction limited points to the sample plane, we descan and pass through our pinhole plate, so now we've removed the outer focus light as you do in any kind of classic array scanning confocal. And now we reassign the image information to where it originated with an emission microlens array, which is isolated just in the emission path, and rescan to the camera. And all we need to do on the camera is integrate, set our exposure time, and we generate our image with high resolution. There's other implementations of analog image scanning microscopy. One that you may have heard of is the SORA, which was developed by Yokogawa. The issue with the SORA is obviously you have two disks rotating at 10,000 RPM, so you can't have another disk in a conjugate plane which is in perfect sync with this disk. It's just, you would need some kind of Chinook style gearing system, which just isn't practical. So in this implementation, they had to put the reassignment optic on the pinhole disk, which means it's also in the excitation path. This has a few issues in that, as you're reassigning the emission, you're doubling the NA back while you do the reverse on the way in, so you actually decrease NA, which is shown here. So in your excitation, you decrease the numerical aperture of the excitation points. The pinholes appear a different size at the sample plane as well. So to resolve that, what you have to do is add intermediary mag. When you add intermediary mag, you effectively project your image onto more pixels. In any live cell imaging application or any camera-based imaging applications, read noise is always the killer. Read noise is always your enemy. And if you have more pixels per unit area, you significantly increase your read noise. The other issue is that, because you have your microlens array this side of the pinhole array, the sample side of the pinhole array, then you'll collect a lot more out of focus light, which could be focused through the pinhole disk. Rather than separate out the out of focus and then do the reassignment, you actually will collect a lot of additional light from the background, because ultimately microlenses, whether they're operating in the telecoms industry, whether they're operating in camera technology, in imaging technology, they basically are there to capture more light. It's just a simple graphical implementation. If you have 10 times as many pixels, you have 10 times the read noise, and your impact to signal to noise is significant. This is a study done at the, because we're shrinking the emission point spread function into a smaller area, as well as the higher resolution, we should be able to increase signal to noise. And this was a study done at the Chinese Academy of Science, where we had the iSIM compared against a regular non-SORA spinning disk in X1, and we actually proved that we could achieve the higher signal to noise as well as achieving the higher resolution. But what does this all mean? This means nothing if it doesn't have a real-world application to meet. And this is just one example from the Hubrecht Institute in the Netherlands. I have to say, so I'm an engineer and physicist. I'm not a biologist, so I end up just reading this bit. So it's U2OS cells with MNU. I think you can probably read for yourselves there. But I think the interesting thing here was they were looking at these bilobe kinetochores during cell division, where the separation they needed to measure was 150 nanometers. The samples were very sensitive, and they needed to image over 70 minutes. And so that was that perfect combination of, and also actually they needed to image two colors simultaneously, because we can do multicolor imaging simultaneously. And it was that kind of wonderful mix of needing the resolution, needing the live cell imaging capabilities, needing the flexibility of the system. And this was a study that they did against the SORA technology. So here when we see the photo bleaching, the SORA kind of bleached out here, while the iSIM really showed very little bleaching. A key point to this, we actually took 34 extra images on the iSIM at each time point, because the SORA just couldn't. The only way they could actually get anything out of it was set like this, but with the iSIM we could just meet the exact needs of the application. So now just a few videos to finish off. So this is from the Gomez lab, just down the road at IMM, so thank you very much for this. I haven't seen Raquel, and I was kind of hoping she could help me describe this sample. Oh, she's sick. Oh, dear, I didn't realize it. Okay. Come and see me, and I'll try and find out to give you some more information on what these kind of cool videos are. But yeah, thank you to Raquel and to the Gomez lab for sharing them. This is a huge thank you to the Gutierrez lab at the Francis Crick Institute. they did give me a little blurb to give. So this protease leakage from damaged lysosomes in the proximity of mitochondria. Publication is referenced there, so thank you to that group. This is from Dylan Burnett at Vanderbilt University, just mitochondria, some actin filaments, ERB, usual nice things to image. From the groups at the Waller Cellular Imaging Center at King's College, look at neural growth cone. Finally, thank you.